The spring shutdown was very frightening. A very small virus, wreaking so much havoc on our world, shutting down economies. All the other labs around us had shut. And then going into my lab, and people have been working harder than they ever worked before.
I don't think I've ever seen across the globe such carnage.
Without the HIV research, there would not be the same COVID-19 response today.
You were basically sleeping maybe 4 hours a night or less.
The immediacy, the need for a COVID-19 vaccine is nothing like I've ever seen before.
There was no option to fail, and that was the one thing that kept us going every day.
Now, if you look at this rather complicated slide of a number of emerging and re-emerging diseases, some of them, every once in a while, evolve into a major public health threat. And others, relatively speaking, are little blips on the radar screen in that they stay confined to the time and the place when they initially emerge. With SARS, we know for sure that we're not dealing with just a blip on the radar screen. But as Dr. Gerberding says, we're not really sure where it's going to go because we are truly in the middle of the evolution of an epidemic. We just don't know whether it's going to peak and then go down or whether it will go down and then come back up at a different season. And for that reason, we have to take this very, very seriously.
The fact that SARS-CoV-2 is less pathogenic than the original SARS actually, I think, has made it more of a global pandemic. For the original SARS in 2003, most people got really, really sick. And the key thing is that people were not contagious until they were symptomatic. So traditional public health measures of isolation and quarantine were very effective. You just had to quarantine people who had fevers and who felt sick, and then you were able to stop the pandemic in its tracks. For COVID-19, it was a completely different situation because it was less pathogenic. Many people only had mild illness, and some people had no symptoms whatsoever. And people were contagious even who did not feel sick. The traditional public health measures were much less effective for COVID-19 than they were for the original SARS. I gave my first talk on COVID-19 sometime in February, and I had a slide that said that nothing else is going to be able to stop this pandemic other than a vaccine. I'm Dan Baruch. I'm the director of the Center for Virology and Vaccine Research at Beth Israel Deaconess Medical Center. I'm a professor of medicine at Harvard Medical School, and I'm a steering committee member at the Ragon Institute. By the end of January, there were two very important conversations that I had. I contacted Dr. Johan van Hoof, who's the head of vaccines at Johnson & Johnson. And I said, I think we might need a vaccine for this. He said his team already started to do work on this as well. Very quickly, we had a signed collaboration agreement with J&J. I also received a call towards the end of January from Dr. Bruce Walker
So my name is Bruce Walker. I'm the director of the Ragon Institute of MGH, MIT, and Harvard. I'm a physician. I'm also a researcher, and I've spent most of my career working on HIV. Each January, I teach a course in South Africa to MIT and Harvard undergraduates called Evolution of an Epidemic, and it's about HIV. And the point is to try and explain both the science behind an epidemic, but also how policy and advocacy influence the course of a pandemic. Interestingly, one of the students from MIT was from Wuhan. She started getting text messages from her family saying that there was this new pneumonia that was just ravaging the city. But we were getting a lot of unfiltered information directly from Wuhan. And this, to me, had all the markings of a global pandemic. And so that as I was flying back from South Africa at the end of the course, I just thought, we have to get involved in this. This is what the Ragon Institute was built for. Viruses have so few genes that they're actually not able to replicate on their own. They just don't have the machinery to do that. What they have is an ability to take over other genes and force them to do their bidding. SARS has 10 genes. E. coli has about 5,000 genes. The human genome is about 20,000 genes. What's extraordinary is that these 10 genes of SARS can basically take over the 20,000 genes that the human has and force the body to start making viruses rather than cellular proteins.
It's just wild to think that one little virus, one little package of genetic material can stop the entire world.
Gilead, I would say, is sort of a firecracker.
Okay, that's totally wrong. I do it again. My name is Gilead Alter, group leader at the Ragon Institute of MGH, MIT, and Harvard, and professor of medicine at Harvard Medical School.
Gilead is one of the most spectacular people I think I've ever met.
We had already been working on HIV, tuberculosis, malaria, influenza, Ebola, you name it. If there was a pathogen that caused problems in vaccine design, you'd be guaranteed that I'd be there.
I don't really know how she keeps so many irons in the fire at the same time, but she manages.
The last, 15 years of my life have been spent on building technologies to understand how antibodies fight disease.
Dr. Alter has experimental platforms in her laboratory to evaluate and to probe antibody responses at a level of detail that's really unprecedented. So as soon as we started the vaccine development of activities, then I talked with Gilead and I said, would you like to work with us on this? And she said yes immediately.
It was a difficult time to work as everything was shut down, trying to decide who could come into the lab to help out with this pandemic response was the first obstacle. But there were a number of volunteers, individuals who just really felt passionate and wanted to be part of the solution. We were basically sleeping maybe 4 hours a night or less, basically trying to do, all the work that we had to get done, to analyze, to put out the data, to communicate it, to help. But then figuring out how to basically get the samples from Dan's lab. There were other reagents within the Harvard system that we had to access, as well as deliver plasma to the Boston Needle. I didn't feel comfortable asking anybody else in the lab to do all of this moving around. So I would get in my car every morning at 8 a.m. I would drive to one laboratory, pick everything up, drive it to another laboratory. And I was driving back and forth with plasma samples, virus, antibodies, and it basically just went like this for months. I mean, I was, the kids in the back seat because my husband was working clinically, and it was the only way to move the science as fast as it needed to move.
The current response to COVID-19 is so intimately connected with our own HIV research over the years. Without the HIV research, there would not be the same COVID-19 response today.
It was 1980. I had just finished medical school and started my internship at Mass General Hospital. I was on call in the emergency room and I saw the patient, a young 19-year-old, who was really emaciated and looked to have quite advanced disease. We thought it was TB. We got an incredible surprise as we started looking into what was going on, that in fact it wasn't TB. It was about 3 different cancers and about 3 different infections, all potentially lethal, all at the same time. Something that the doctors at MGH had never seen before. They said, you'll probably never see another case like this, and the patient rapidly died. And a couple of weeks later, we saw another case just like it. I mean, that was clearly a pivotal moment for me, and it really changed my whole perspective on what I wanted to do with my career. Become not just a physician, but also a scientist, to try and learn from the patients that I was seeing to see if we could ultimately conquer what appeared to be an entirely new disease.
AIDS, or acquired immune deficiency syndrome, kills 4 out of 10 people who acquire it, and that mortality rate may climb. The incidence of AIDS has doubled every 6 months. Fear of the disease is itself becoming epidemic.
HIV is one of the defining pandemics of our generation. It's not going away anytime soon. When I started my lab in the early 2000s, it was one of the leading causes of death.
It's killed over 32 million people since it first was detected, and we still don't have a vaccine for it.
HIV has scientific challenges that are unprecedented in the history of vaccinology. HIV has a vast sequence diversity, orders of magnitude more than what we see for COVID-19.
HIV mutates extraordinarily rapidly. When we make copies of our proteins, the rate of mistakes we make is 10,000 times lower than the rate of mistakes that HIV makes. HIV also replicates rather quickly in a person, making 1 billion virus particles per day. Most of the viruses are junk because they're mutated. But you see, even in a particular person, the virus mutates so quickly that it beats our immune system.
Another problem is that it integrates into the host chromosome. COVID-19 cannot do that. So HIV is able to hide away from the effect of drugs and the effect of the immune system, and then pop up later when you stop drugs. And so it becomes a lifelong infection very rapidly. I had been working on HIV really my entire career. I think the depressing, really low moments came with one after another vaccine failure. Is it really a question of money, or is it a question of ideas? And that's where we started thinking the way that science is set up with these sort of specified disciplines and a siloed nature, it was very hard for somebody who wasn't already working on HIV to get into the field. If you could bring people from other disciplines, from engineers and physicists, mathematicians and computer science people and biologists, and it's putting them together and having an environment where they're talking to each other, where people are bouncing ideas off of each other.
I'm president emerita at MIT. I was president from 2004 to 2012. What I'd say about the ambition to bring MGH together with other parts of Harvard and MIT was pretty audacious. But we've got a lot of barriers that make it hard for people to find their way out of your own department to work with a colleague in another department and a different institution. I mean, the barriers just get greater and greater.
If you ask me, did I realistically think we could achieve that vision, I would have to say, no, I didn't think that.
One of the most remarkable evolutions within our immune system that occurred sometime when we transferred from fish to jawed fish, we developed the ability to evolve immune responses. So what we essentially did is we created parts of our genomes that could rapidly rearrange. Now that combinatorial genomics is something that really only occurs in T cells and in B cells. Those are two cell types that are so critical for fighting viruses and pathogens. So an antibody is a protein that is generated by B cells, and these are the proteins that essentially act as magic bullets. They are shot all around the system, they float around in the blood, they go throughout the tissues, and they latch on to the surface of the pathogen or of the infected cell, and they essentially signal to the system that something is aberrant about whatever it is that they're attached to. And they provide instructions on how the immune system should clear or destroy anything to which that antibody is bound to. One of the issues was that when we started developing the antibody assays, I couldn't produce protein fast enough. I have to say that some of the SARS-CoV-2 proteins are not the easiest proteins to make. The quality of that protein will dictate the quality of the data that comes out. And that was just really the essential first catalytic piece of the puzzle that we needed to basically get all the experimental work up and running.
I'm Aaron Schmidt. I'm an assistant professor of microbiology at Harvard Medical School and at the Ragon Institute of MGH, MIT, and Harvard. We're a group of structural biologists primarily and protein engineers. One of my graduate students, Jared Feldman, came into my office very early on in the pandemic, and he asked and said, should we be working on this?
And you're here in our lab.
And I said no, and I didn't kick him out of my office, but I basically said, this is not something that we need to divert our attention to. There are other experts that should be taking care of this, and let's focus on what we've been working on for the last couple of years. 2 days after, Galit, with her enthusiasm, coming into my office and saying, we need these proteins.
Aaron volunteered.
We need you to work on this with us. Can you make them for our efforts? So I had to, with the tail between my legs, call Jared back into my office and say, all right, it looks like we're going to start working on this. What's interesting about it is that, so the SARS coronavirus, the image that I think all of us see is that gray sphere with the red spikes. So that is the spike protein, and that's the protein that we and others work on. And what we have done is to take that spike protein, and there's a small little part of it called the receptor binding domain, and that's what's responsible for engaging a target cell for the virus to then bind to and enter in and begin its replication. That's what we're recombinantly expressing and giving to other individuals like Dan and Galit. And that protein is what people use in their assays to then understand the immune response that is elicited either by natural infection or by a vaccine candidate. 3 of my graduate students, Jared, Blake, and Tim, started to work on it and start to clone and produce these proteins.
And he shared those freely and willingly with everybody who wanted to do the science.
Galit had spread the word that we were making these proteins. So we started getting request after request after request. I think we've produced over 400 milligrams worth of these proteins and shipped to 30 to 40 different labs across the country and even across the world. The 3 of them that came in during the shutdown and arguably risked their lives to be able to do these purifications and production for Galit and Dan and others' efforts to try to develop a vaccine. They're the true heroes of this whole process.
An assay is essentially a recipe for doing a scientific experiment, for measuring something that is essential to an immune response. So the first thing we have to do is we have to trap the antibodies that we want to measure. And the way that we do that is by capturing protein on a plate, putting plasma on top, The antibodies that are specific to that protein will bind. And then we wash away everything else. And so all we're left with are the antibodies that recognize SARS-CoV-2. Then we can measure those with different types of detections. And this information allows us to not only figure out how much antibodies somebody has in their blood, but also allows us to interrogate the quality of those antibodies.
Bruce has always been known to me as a slightly more senior, very distinguished scientist and physician. His exploits and efforts to confront HIV, especially in South Africa, have been exemplary, extraordinary. He's laid the foundations for better treatments and ultimately for an understanding of how we might one day control HIV with vaccines. And then, of course, he'll be legendary for his founding of the Ragon Institute.
Yeah, this is really quite an unanticipated and to me, kind of remarkable sequence of events. We were working in South Africa and had been for a number of years. We had formed a collaboration with a mission hospital there because it was the mission hospital where we were actually able to treat patients. This very visionary leader of the mission hospital had implemented an electronic medical record to keep track of patients. The superintendent of the hospital said, would I meet with the owner of the EMR to tell him that it was saving patients' lives?
I'm Terry Ragon. I run a computer software company. People were telling me that there's this person I should meet, Bruce Walker. And people were apparently telling him also that he should come meet with me.
When I came back to Boston, I went over to meet with him. Again, I knew nothing about him or his company.
And right away he blurts out, I don't have a clue why I'm here. And I said, well, you know, I really don't have a clue why you're here either.
And he started asking me really good questions about what it was that we were doing in South Africa, why his EMR was saving lives, and also, importantly, why we were trying to build research institutes in South Africa when, in fact, we had everything we could possibly want at Harvard and MIT. So I also said to Terry, if you want to join me, I go over about every 2 months.
So his next trip was 2 weeks later, and I went on the trip with him. So I actually told him early on, just to take the pressure off, it's unlikely I'm going to give you any money. And I'm sure at this point he's thinking, well, this is going to be a wasted trip, but he had other things to do. But it also freed me up to have the opportunity, instead of just being shown around, to look around myself. It was pretty intense. I saw Bruce and I said, you know, Bruce, there's so much money already in HIV that any money that I provided would be just a waste of money. There's really nothing I can do until somebody does something about HIV because it has so overwhelmed these hospitals, the social services, everything. And Bruce's exact words were, let's not be hasty.
And I said, you know, what I'd really like to do is I'd like to solve this problem. I'd like us to make an HIV vaccine. And I think the scientific community can do that. It's just that we're not applying all the knowledge that's available to be able to do that.
When he came back, he was just really excited about it. Just felt that, you know, there's a lot of work being done separately, like Harvard, MIT, Mass General. They were all working independent of each other.
Terry's response to that was, wow, that's clearly a big problem, so it's going to take a long time. And you probably then need about $10 million a year for about 10 years. Is that about right? And I said, yeah, that's about right. And he said, well, my wife and I would like to give that to you. And it was just this incredible out-of-body experience where I was trying to rewind what had just been said and thinking, did I really hear what he just said? And that was really the beginning of the Ragon Institute.
You know, really there was, you know, two goals initially. You know, one was, of course, a vaccine, and I would always say cure. for HIV. But the other was always the belief that if we're successful, we'll learn so much about the immune system that we ought to be able to cure a wide range of diseases at the same time. And that's turning out to be absolutely accurate. And certainly that's the case here.
The human history is inextricably linked to infectious disease-causing pandemics. But because of better sanitation, vaccines, and frankly, good luck, it had been more than a century since we had seen a pandemic. My name is Arup Chakraborty. I am a professor at MIT, and I'm also one of the founding members of the Ragon Institute. The earliest vaccines, before we knew about pathogens or the immune system, our ancestors noticed that someone who recovered from smallpox didn't get sick again. And so they could be caregivers. They took that one more step. They started taking scabs from smallpox patients, powder them, and then administer it in two different ways. The Chinese and the Indians did it differently. So there you were giving a live virus. So many people just died upon vaccination. And then came the next big deal, which was Jenner, where Jenner understood, serendipitously, he still didn't know about pathogens or immunity, that you could protect a person from smallpox by injecting a related pathogen that was harmless for the human. And that was cowpox. And that's the vaccine that eradicated smallpox from the world. Then in the 18th and 19th century, Pasteur and Koch understood, hey, you didn't have to go look for another pathogen that was related, because that's hard. You could just inactivate or kill this pathogen just right, so that if it was killed or attenuated, it couldn't cause disease, but it was still more or less like the pathogen, so you'd get an immune response to that. And then came the 20th century, when we had recombinant DNA technology, so we could learn how to make proteins. So rather than giving the whole bug to improve safety, start making a lot of the protein and injecting that. In the 21st century vaccines, what we are injecting is not even the protein of the viruses. What we are doing is just giving our body information about the proteins of the virus. And how do we give that information? One category of these 21st century vaccines are RNA vaccines. Moderna and Pfizer BioNTech are examples.
The mRNA platform uses something called messenger RNA, which is the messenger inside your cells to make your cells make spike protein, which your body recognizes. mRNA was a very untested technology. It had been used really only in clinical trials.
And they can tweak the program, so this is one way that they say they can address variants pretty quickly. I mean, Moderna started working on the vaccine and distributed the first batch to the government in something like 63 days. You know, that's astonishing. But there are all different types of technologies. There are vaccines that use another virus as a vector to deliver it.
And then there's the Johnson & Johnson vaccine. It uses a vector called Ad26.
I started a project when I was first-year medical student, and I decided I wanted to make an HIV vaccine. It seemed like a very reasonable project to do. Now, it couldn't be done in the summer. And one could argue I'm actually still doing that project today.
When we formed the Ragon Institute, we brought together a number of scientists from Harvard and MIT and MGH and others in this incredible biomedical research ecosystem here in Boston and Cambridge. One of those was Dan Baruch, who was working in the early stages of a vaccine for HIV. Dan became one of the founding members, and we supported the development of that vaccine. Dan is a very clear thinker. He's extremely motivated. I think he understands vaccine development better than any other academic investigator.
He's a smart character, no doubt. But he's also got that intensity, that drive, which the best scientists have.
I've been leading my own research group here at Beth Israel Deaconess Medical Center since 2004. And during those years, one of my first projects was to develop some new vaccine platform technologies. So adenovirus serotype 26 is a common cold virus. So we deactivated them so they don't cause disease. The purpose of a common cold virus is essentially to shuttle a piece of another virus into cells. And it turns out common cold viruses are very, very good at doing one thing, which is getting into human cells. And we were able to stitch in different pieces of viruses of interest. We can take an antigen of interest, in this case, the COVID-19 spike protein. We can take the DNA and we can insert the COVID-19 spike protein DNA into the adenovirus vector. And once it's in cells, the COVID-19 spike protein is made and the body makes an immune response to it.
I think we are working with Dan Brook more than 10 years. It was very simple and fast to say, let's bundle the power of our science.
Baruch and his team took some extra steps in testing different versions of their vaccine before deciding which one to go forward with.
So for the COVID-19 vaccine, we looked at a variety of different forms of the spike protein in collaboration with our partners at J&J. We looked at full-length spike protein, just pieces of it, as well as modifications of it. And we settled on initially a version of a full-length spike protein that also had stabilizing mutations.
I think that was very smart from the scientists to do that, because it turned out that there was a lot of difference between different vectors in the challenge models in animals. And when that came out, I say, yes, this is going to work, yeah? Full protection against disease, almost full protection against transmission, this is going to work. And that with a single dose.
We got very good news over the weekend. The Food and Drug Administration issued an emergency use authorization for a third safe and effective vaccine, the Johnson & Johnson vaccine, which will help us defeat the pandemic. While we have much work ahead of us on so many fronts, this is certainly a very encouraging development.
The trial showed in the United States a 72% effectiveness in preventing mild illness, 66% throughout the world, largely driven by the fact that there was reduced efficacy in South Africa because of the resistant viral variants. But the most important results from the trial are that we showed that the vaccine provided 85% protection against severe disease and 100% protection against hospitalization and death. That was true not only in the United States, but also in Brazil and Latin America and in South Africa.
The J&J vaccine, a number of scientists were championing it as the potential workhorse for developing countries, largely because of kind of logistical challenges with the other vaccines. Both the Pfizer and Moderna vaccines have to be kept in very cold storage. Then, of course, the other huge advantage that people were touting with the J&J vaccine is it was one shot, not two. And in poor countries and in rural areas, a one-and-done vaccine is a big, big plus.
Vaccinologists talk about the world's vaccine, the vaccine that you could give to everyone, and it would be effective and it would be safe. And the J&J vaccine has a lot of those qualities.
And we are working with Gavi, COVAX, we're working with the African Union, with PAHO, with UNICEF, on how can we maximize now the access to the vaccine in the developing world.
One of the only problems with it has been that production hasn't ramped up as quickly as was expected. It's been hard to make.
Very early on in the process, already we started working with Emergent in the US to do the vaccine production for us on the ground in the US. At a certain point, there were some quality issues at Emergent, and we quickly worked with the regulators to find the way to correct for that, as well as restart manufacturing.
You know, even if you develop the world's best vaccine, it is an enormous logistical challenge to put it in hundreds of millions of arms, let alone billions of arms.
You don't know what's going to happen when you put it into millions of people. When millions of people have received something, there could be rare side effects that would never show up in any clinical trial.
As we speak, the FDA and the CDC has announced that they are now recommending a pause in the use of Johnson & Johnson's vaccine.
This morning, the U.S. is calling for an immediate pause on the use of the Johnson & Johnson vaccine. Jerry Willis now with more details.
The rollout of the Johnson & Johnson vaccine is facing a couple of hurdles at once. States are bracing for a nearly 85% drop in doses next week.
There were the unexpected cases of very rare but severe blood clots seen in a small number of individuals, approximately the current estimates about 2 or 3 in a million. And that did lead to a 10-day pause of administration of this vaccine.
Now, the pauses are expected. The pauses are necessary because we're not in a fully licensed vaccine product space. We're in an emergency use authorization stage of vaccine development. So my laboratory does look at side effects. We're very interested in the entire immune response, not just to the vaccine itself, but really as a holistic response within the body to different types of inflammatory cues. And so we have had a chance to work on some of these thrombosis side effects that we're seeing in following the J&J vaccine, but also looking at the potential cause for anaphylaxis following some of the mRNA vaccines that we've seen pop up also in the media.
If you introduce new vaccines or new medicines to millions of people, you will learn new things. And the mRNAs have different side effects. We learn about that, we warn about it, and we make sure physicians know how to treat it and to be. But that is not abnormal with vaccines. You always have some kind of side effect.
So it seemed like a totally legitimate thing because the last thing you want to do is to stick with something and kind of blindingly ignore some legitimate concerns. So they paused it for 10 days, and then they did, which is what medicine does all the time, which is they did a kind of a risk-benefit analysis, and they determined that the benefits far outweighed the risks.
Within just 10 days, they accumulated a huge amount of knowledge. They concluded that there were only a few additional cases, not hundreds or thousands of additional cases. They also did a very careful risk-benefit analysis, really to demonstrate that the benefits of the vaccine vastly outweigh the risks in all subpopulations. And so that was very valuable.
And when I heard that incident, you know, for me, I thought, gosh, that's about the most fabulous drug you could ever make, because there are very few drugs that have an unfortunate side effect in 1.5 million doses. So this idea of risk is misunderstood by the population.
I think people are concerned about the speed with which this happened. It's not because corners were cut, it's because there were existing technologies that allowed those to be immediately applied.
There are no piece of the vaccine development pipeline that have been deleted. Everything has now been built into this contracted pipeline that allows us to still balance safety with speed. In this particular pandemic, there was no choice. Everybody had to basically commit to making a vaccine.
If you commit to making a vaccine for pandemic, you have to do everything in parallel. The laboratory work was done, the animal work was done, clinical work was prepared already. You don't start the 45,000 study overnight. That was already in the making months on beforehand. The same with manufacturing. As soon as we knew this is going to be global, massive need, we said, okay, we need at least 2, if not 3 drug substance facilities, with the goal to make 1 billion vaccines this year, and more than that next year. Huge investment, but okay.
You know, this is a very, very tough problem, vaccine hesitancy.
Well, it's really important to realize that there's a spectrum of reasons why people decide not to get a vaccine. There are all sorts of barriers in the way of people getting the vaccine. There are also a lot of people who can't take time off from jobs, and then there are people who have not been served well by the healthcare system as it is. And people have long memories about mistreatment.
But most are hesitant to get a vaccine because they don't believe the science. Science skepticism is the larger name for this. I think that we have a really serious communication problem. There are communication media that allow everyone's ideas and everyone's point of view and everyone's opinions to have equal weight.
We are saying we expect more from our technology companies. We're asking them to operate with greater transparency and accountability. We're asking them to monitor misinformation more closely. We're asking them to consistently take action against misinformation super spreaders on their platforms.
There's about 12 people who are producing 65% of anti-vaccine misinformation on social media platforms. All of them remain active on Facebook, despite some even being banned on other platforms, including Facebook, ones that Facebook owns.
Well, I remember when my daughter was born 22 years ago, there were all these reports about that there might be some link between vaccines and autism. There was a study in The Lancet that turned out to be pure rubbish and was retracted. That did a huge amount of damage. But a lot of people have cleaved to these misperceptions. I mean, if you talk to some of these vaccine authorities, as I have, they would argue that vaccines may well have had the biggest impact on the health of the world of any medical advance in human history.
My faith in science as a process has been strengthened. I mean, the vaccines are a great example of the fact that you can figure this stuff out.
We're certainly seeing cases drop dramatically, particularly in places with high vaccination rates. It's very hard to get from real-world evidence data, from the kind of data we collect, information about exactly how good one or another vaccine is once we're out of the clinical trial process. But it's been crystal clear that these vaccines have worked, and we've seen all of them showing efficacy in the US and in other countries.
But we cannot be complacent here. The virus is not yet gone from the world.
My wife's brother lives in India. He is a doctor. He runs all the COVID wards in one of the major hospitals in New Delhi. And what he has described to me in the rare moments where he's available is that what we are seeing there is just human carnage.
Well, I think if you look in the world, you'll see that there are very different levels of prevalence of infection and ongoing levels of new infections in different countries. This is an evolutionary arms race between the virus and humans. The virus makes mistakes as it reproduces itself. So for every person in the world that gets infected, there's an additional chance for the virus to find a mutation that will render the immune system less able to target it.
The Delta variant, first identified in India, has now spread to nearly every state in the U.S.
Importantly, I hope that we can equitably distribute vaccine, particularly when you think about this as this is a global problem.
This is small planet Earth. We're one community. And we've been devastated as one community. We need to work together as one community in the future to be more effective.
Well, right now we're doing a lot of work with the viral variants. We're exploring responses for all the vaccines against viral variants. We're also examining whether current vaccines will be protective in animal models with variant challenges.
But I'm also worried about the next coronavirus pandemic. The first two lethal coronaviruses were much more lethal than the current one, but they were much less transmissible. What I'm worried about is a coronavirus SARS-CoV-3 that has the transmissibility of SARS-CoV-2, but the lethality of SARS-CoV-1 and MERS. If you think about that, that is really an existential threat to humanity.
There's so much technology that grew out of HIV There was this massive investment, so much effort, and so many HIV researchers have all been redeployed to COVID. And I think that's something that it's like totally wild. Like if you look at who's running the COVID trials, who's writing all the papers, it's almost always HIV researchers. So for us, it was a natural transition to SARS-CoV-2 when it emerged.
And we spent over 15 years in our projects to try to develop an HIV vaccine. And right now, with our partners at Johnson & Johnson, as well as the NIH and the HVTN, we have 2 large ongoing studies that both are aimed at looking at the efficacy of this vaccine. Currently, this is the only HIV vaccine that is in large-scale phase 3 clinical trials.
In my opinion, anything above 50% activity could curb the HIV infection in the world. And so it also would be the first proof of concept that an HIV vaccine can work.
If the HIV vaccine is effective, then it will have a huge impact on the world. HIV has reshaped societies and has reshaped economies and has caused immeasurable stress on the healthcare system.
When HIV cases were first identified, we didn't know what caused them. It took 4 years then to actually even figure out that it was a virus that was causing this new disease, and it took another year to develop a diagnostic test. Those are steps that took years for HIV. Those same steps took less than 2 months for SARS-CoV-2. That's just a spectacular achievement by science that I hope people recognize. And it's built on 40 years of additional experience and the development of these platforms that can be rapidly switched out for new pathogens as they arrive.
But I do think that the work on COVID vaccines will back inform HIV vaccines and vaccinology in general in many ways. It shows what is possible when there is a pressing global need, when there is a passion to actually move a program forward, and when there is resources to do so.
It is remarkable how quickly everybody collaborated with one another and shared information and shared resources with one another. And I think that absolutely helped pave the way for where we are today.
As soon as the virus was sequenced, the Chinese released that to the world. That meant that all these people with vaccine platforms could immediately start working.
Scientists are working together in ways that was not fathomable before this pandemic ever occurred. No one is working on their own to solve these problems. And all this technology and all of this openness that has come about from this pandemic is likely to impact other fields, including cancer, autoimmunity.
We've put together a community which is building an infrastructure that I think is going to not only allow us to confront the current pandemic, but create the kind of capabilities that we need to be ready for the next one.
To participate in the creation of the Ragon Institute, which to my mind is an unrivaled collaboration. And it's not just that the science is great and the medical interventions are great, but they've created a community of hope, community of purpose, and in COVID times and before COVID times, it is not common to find communities that so raise up so many people to do things that improve the world.
I think when historians look back on the COVID-19 pandemic, there will be a lot of terrible things remembered. But I think there will be one thing that will be bright and shining through, and that is the power of science.